everyone. In this video, we're going to begin considering the physiologic processes needed for male reproduction. So we're going to start off by talking a little bit about the formation of the sperm cells. And in doing so, I just want to revisit a couple of vocabulary words and sort of make sure that we're all on the same page. So previously we described mitosis, which is basically the reproduction of body cells or somatic cells. So when we think about reproducing individual cells, we just want to make more of them. We start with 23 pairs of chromosomes as far as the DNA is concerned. And when we're finished with reproduction, we end with 2 cells that each have 23 pairs of chromosomes. So 46 chromosomes total in the beginning and 46 at the end in each cell. When we talk about reproduction of a person, or making more people, we also include the concept of meiosis, which is basically 2 cycles of cell division that results in 4 cells that are 23 chromosomes each. And the 23 chromosomes each, they're not paired. It's just 23 individual chromosomes. And these cells are called gametes or sex cells. And so for the male, those would be the sperm cells. The other thing we want to mention is gonads, which in the case of the male are the testes, which are basically endocrine organs that produce hormones and also, of course, produce the sperm. So first, let's begin by considering the testes and taking a look at all of the structures that are present inside that we need for the production of sperm. And so we're going to do this in a couple of stages. So this is sort of a slice open of the testes, and what we can see is coiled structures called seminiferous tubules. And the seminiferous tubules are the actual site of sperm production, and it's difficult to tell from this picture. But the seminiferous tubules total about 80 centimeters in length, and if we were to stretch them all out and consider the amount in both testes, we would have about a half a mile worth of seminiferous tubules. So it really is, even though they're densely packed, it really is a ton. And so as the sperm cells are produced, they actually move out of the seminiferous tubules into the rete testes and then the efferent ductules, which are basically just there's cilia and stuff that help sort of move the sperm out and then into the epididymis, and then they'll eventually exit through the vas deferens. A couple of other things to mention is that the testes are housed in the scrotum, which is stored outside of the body. So the testes are actually outside the body, which is important because it allows the sperm cells to grow at a temperature that's about 2 degrees cooler than normal body temperature. We also have in sort of around the testes in the scrotum the cremaster muscle, which is important because it allows for a little bit more temperature control. So if the temperature outside is extremely cold, then the muscles will contract and pull the testes closer to the body. And if it's really warm, they'll relax and allow the testes to be further away. So it helps to stabilize the body temperature. Now let's take a look at the seminiferous tubules. If we were to sort of cut a slice down the middle, what we can see is some Sertoli cells, which are also called sustentacular cells, that essentially make a ring around the to form the exterior of the tube. So basically, these are a form of epithelial cells, and their job is to nourish the developing sperm cells. And then also what we can see is that there are tight junctions between the cells. So this actually forms the blood-testes barrier. And this is important because it makes sure that there's no pathogens or anything that can access the developing sperm cells. So they're sort of protected from disease in that way. Now, as we notice the sperm in purple, there are younger sperm that are closer to the exterior, and then we have the older sperm closer to the center or the lumen. So essentially, as several divisions occur to make the sperm cells, we see that they move inward and then they're able to travel. Surrounding the seminiferous tubules, we have basically a connective tissue space, and in here we have Leydig cells, which are also called interstitial cells or nurse cells. Their primary function is to make testosterone. And then, of course, we also have a vast blood supply, which is important. And then finally, it's basically loose connective tissue that sits around them. So what I want to do next is talk about spermatogenesis, which is basically the cell divisions, a series of cell divisions that are needed for the production of the sperm cells. So in the context of this, I just want to mention that we'll draw a pair of chromosomes, and we'll sort of show one that's filled in and then one that is just with some lines through it. And the one that's filled in will represent the dad chromosome, and the one that's just lines will represent the mom chromosome. And this will help us to keep track of the different divisions. So spermatogenesis is basically 3 steps: mitosis, followed by meiosis 1, followed by meiosis 2. And we're just going to draw each stage and sort of take a look at what happens so that we can understand how sperm cells are produced. So first we notice that we have a spermatogonia, which is a stem cell. And in this stem cell, we've drawn 3 pairs of chromosomes. So it would be really impractical to try and draw all 23 pairs, even though that's what's actually there. But we're going to start with just 3 and just watch what happens. So what we can see is this is basic mitosis. So we're going to replicate all the chromosomes, and then in the end, and the dotted line in the middle is our sort of transition state. So it's the cell after the replication of the chromosomes before division. And then what we end up with is 2 cells that look exactly like the original, and they both have 23 pairs of chromosomes. We can see here are 3 pairs. One of the cells is a spermatogonia, or a stem cell, and the other one is a primary spermatocyte. So if we were to take the stem cell and divide into 2 primary spermatocytes, then we would have a finite number of stem cells, and then we could make a finite number of sperm cells. And so because we don't want to do that, every time we do mitosis, one of the cells will differentiate into a daughter cell that will become a sperm cell, and one of them will remain a stem cell so that we could reuse it over and over again. So this initial division, we notice an exact copy, 1 dad chromosome and 1 mom chromosome for each new cell. The primary spermatocyte will then divide again, and so as we take the primary spermatocyte and convert it, we're going to be in meiosis 1, and we can see first in the transition state we have this replication of the chromosomes again. So for every original pair of chromosomes, we now have 2 copies of dad and 2 copies of mom. The 4 together are all connected. They sort of essentially hold hands during this time, and that's called a tetrad. And during the time where we have the tetrad, we can have this phenomenon called crossing over, which basically means that it's almost like if you imagine like 2 people like laying next to each other, and then they just sort of like cross their legs over each other, and then in this case like switched legs. That's kind of what happens. So a piece of the chromosome can switch places with another, and this is important because it allows for genetic variation. So why it's possible to have so many different arrangements of characteristics that come from mom and dad. So from here we'll get our next division, and this time what we're going to see a major difference in comparison to mitosis is that when we form the secondary spermatocytes, we have 2 of them, we have both of the dad chromosomes go to one cell and both of the mom chromosomes go to the other cell. Now let me be really clear about this, because I don't want you to think that one cell contains all 23 of the dad and the other cell contains all 23 of the mom, because that's not necessarily true. It can be any arrangement, but what we do have is you can see both of the, so let's just look at the blue chromosome. You can see both the filled in chromosomes, so those are both dad, go to one secondary spermatocyte, and then both of the non-filled in ones that are mom go to the other one. Now, if we compare and look at the green one, we notice that the mom chromosomes went to that first cell and the dad chromosomes went to the second cell. So it doesn't really matter what arrangement of dad versus mom we have in either cell, but in either case, the original dad and the copy of dad have to stay together, and the original mom and the copy of mom have to stay together. And so the dad and copy go to one and the mom and copy go to the other, regardless of so if chromosome number 1 is, dad to one and then mom to the other, chromosome number 2 could be the reverse. It really doesn't make a difference as long as we have this arrangement of dad in one, mom in the other per chromosome. Now, I just want to point out that if we didn't do it that way, if we had one dad and one mom in each, all we would have done is mitosis, and that's not what happened. Further, just for a little bit of housekeeping in terms of the numbers, when we had our primary spermatocyte, we had 46 chromosomes, 23 pairs. When we look at this transition state, we do have 92 chromosomes, but we would not consider it to be 46 pairs. And so then when we make our secondary spermatocytes, we say that we have 46 chromosomes, but again, we don't have 23 pairs. It's not the same thing as if we did mitosis. Finally, we move to meiosis 2, and essentially now we're just going to take and divide each of the secondary spermatocytes into 2 cells where we get 1 chromosome of each number, 1 through 23, per cell. So these are actually the beginnings of what's going to be the sex cells. So this is our sort of our product, and we call them spermatids. Now, you may be thinking that these really don't look like sperm cells. In order to get them to actually look like sperm cells, we need to undergo another process called spermatogenesis. And it's basically making the shape of the sperm cell that we really recognize. And the official name is spermatozoan for these cells. And we can see that they have heads and tails and are a little bit different. So now let's take a look at the male reproductive anatomy just so that we can follow the path of travel as the sperm cells exit the male body. So obviously this is a side profile slice of the male reproductive anatomy, and I just want to go through and label all the structures so that we can then talk a little bit about the actual path of travel of the sperm cells. So obviously, again, we can see the testes and the epididymis, and I didn't draw in or label the rete testes or the efferent ductules, but we'll know that they're there from the previous drawing. And then we see the vas deferens or the ductus deferens as it travels up kind of into the male body. And I just want to mention that the ductus deferens is actually enclosed in something called the spermatic cord, which also contains blood vessels and nerves. We can also see the urinary bladder and the ureter that's going to deliver the urine to the urinary bladder. There are a couple of different types of vesicles or glands that will secrete things into the path of travel of the sperm so we can kind of make semen. So we have our seminal vesicles, and there's 2 one on each side. We have our prostate, and we can see that the prostate surrounds the beginning of the urethra. We have our bulbourethral gland, which is also called Cowper's gland. And the space sort of before the urethra merges with the ductus deferens is called the ejaculatory duct, and so that's where we get the beginning of our semen and our secretions to sort of mix with the urethra. And we can see that the urethra exits through the penis, and we'll just label the parts. So we have the root, which is the sort of the point of attachment of the penis to the male body. We have the shaft, which is the length of the penis, and then we have the glands, which is the distal portion of the penis that surrounds the external urethral meatus, or the hole where the urine and semen comes out. So before we can follow the sperm out, I just want to draw a more detailed view of what the sperm looks like when it's all grown up. So we can see that there is a head, a neck, a middle piece, and a tail. In the head, we have what's called the acrosomal cap, which contains various enzymes, including something called hyaluronidase for fertilization. We'll kind of talk more about that when we actually talk about fertilization. We also see a nucleus, which contains the 23 chromosomes that we mentioned previously. And we have a middle piece, which contains a mitochondrial spiral. So we have to have production of ATP here so that we can power the movement of the tail. And the tail is actually the flagellum. And this is the only cell in the human body that actually has a flagellum. And it actually powers the forward swimming of the sperm. And so we need ATP in order to make that happen. Okay, so as we make our way out of the seminiferous tubules, I'm just going to highlight a couple of things that happen in each of the different areas. So first we see that we move from the seminiferous tubules to the rete testes to the efferent ductule, which contains cilia to sweep the sperm cells into the epididymis. And just to give you an idea of the timing, it takes about 72 days for the sperm to be developed and move all the way out of the seminiferous tubules. And there's like an interesting cycle where the body starts producing new sperm in different segments of the seminiferous tubules every 16 days or so. Once we get into the epididymis is responsible for a couple of important things. One is maturation. So initially, the sperm cells are very weakly motile. They don't really swim well. And then when they get to the end of the epididymis, they swim quite a bit better, but they only can swim in like the forward direction. The epididymis is able to store the sperm for about a month. It takes about a month to get from one end to the other. And it also will recycle damaged sperm and contribute some secretions to the environment for the seminiferous tubules. As the sperm move into the vas deferens or the ductus deferens, they can actually be stored there for quite a while and then can move out. Before we comment about that, I do want to just mention that if the testes twist around the spermatic cord for some reason, which is basically an occlusion of the blood flow to the testes, we get something called testicular torsion, which is considered a pretty significant emergency for a male. Once we're in the vas deferens and we want to actually physically move the sperm out of the body, we need some type of sympathetic activation. So sort of in conjunction with sexual arousal, we'll get sympathetic activation and that will cause peristalsis in the vas deferens that will propel the sperm forward so that it can meet up with some of the vesicles for secretion. So let's talk a little bit about what happens in terms of adding stuff to the sperm cells to make semen. So the seminal vesicles contribute about 60% of the semen, and they release fructose, which provides the energy that the cells need. They release prostaglandins, which are responsible for helping with smooth muscle contractions, and then fibrinogen, so that the sperm cells and the semen can form a clot inside of the vagina initially upon arrival. The prostate is the next stop, and it contributes about 30% of the semen, which includes things like buffers and antibiotic chemicals so that the male doesn't get a urinary tract infection. And then we can see that as we pass through the prostate and enter the urethra, we have one more gland to pass, which is the bulbar urethral gland, and we get release of sticky mucus, and that also can help to neutralize the acid of the urine if there's any urine left over in the urethra. So when we get to the point of exit, we will have ejaculate, which is about 2 to 5 mLs of semen, and then about 60 to 100 million sperm cells per ml. So that's really quite a bit. And there's a couple different criteria for considering sterility in a male, but if the sperm count is low in this volume, then that can be one way that it could be determined. I want to talk just for a moment about the physiology of erection. So what we can see is now a slice or a cross-section of the penis, and we can draw in, we can see the dorsal vein. There are 3 columns of erectile tissue, 2 corpora cavernosa, corpora being the plural of corpus, and then we have the corpus spongiosum. In the center of the corpora cavernosa, we have arteries, and in the center of the corpus spongiosum, we see the urethra. So among these 3 columns of erectile tissue, what we could say is that normally at rest, the arteries are constricted, and so there really is kind of like limited blood flow to the penis, and so it remains flaccid. But upon sexual arousal and subsequent stimulation, it's actually parasympathetic innervation that's responsible for erection. And so I just want to kind of mention how this happens. When we have parasympathetic stimulation of the erectile tissue, what we get is release of nitric oxide. So NO is released from the neurons. Nitric oxide is actually responsible for generation of the second messenger, which is cyclic GMP. So it increases the concentration of cyclic GMP, and consequently we get vascular smooth muscle cell relaxation. So vascular smooth muscle relaxation leads to vasodilation and then subsequent erection. And as a side note, for those patients who are taking things like Viagra or any other erectile dysfunction medications, it actually prolongs the availability of cyclic GMP, and therefore it will result in prolonged vascular smooth muscle relaxation, prolonged vasodilation, and prolonged erection. The final sort of disease I want to mention in conjunction with male reproduction is priapism, which is basically an erection without sexual arousal. It's commonly associated with spinal cord injury. So if you think about having like a spinal cord transection or something, that damages the spinal cord below the sympathetic neurons, what you're left with is unopposed parasympathetic stimulation, which will result in an undesired erection. Of course, we can't talk about male reproduction without talking about the hypothalamic pituitary testes axis. So we want to just sort of revisit the endocrine control of male reproduction. And so let's just draw in our hypothalamus and anterior pituitary, and then we'll draw in our Sertoli cells, Leydig cells, and blood capillaries that are present near the Leydig cells. So we know that GnRH is released from the hypothalamus, and it triggers the synthesis and release of LH and FSH from the anterior pituitary. The Sertoli cells produce an FSH receptor, and you might remember this because the S in Sertoli, and then there's an S in FSH. And then the Leydig cells possess the LH receptor, and again, L for LH and then L for Leydig. So we're going to kind of jump back and forth here to describe the events, but the Leydig cells are responsible for producing testosterone. We'll abbreviate that as T. And the Sertoli cells make two things: androgen binding protein, or ABP, and inhibin. So first, let's imagine that we have GnRH stimulating LH, which then travels in the blood and arrives at the LH receptor, where it binds and triggers the production of testosterone in the Leydig cells. Some testosterone will move, and of course, testosterone is a steroid, so it's a lipid soluble, so it's going to move from the Leydig cells into the Sertoli cells just by just passive diffusion. When the testosterone enters into the Sertoli cells, the ABP is going to bind the testosterone. So this is important because it allows for the testosterone level to remain high inside the Sertoli cell. I think that it would be easy to think of it very similar to when glucose enters a cell and then immediately binds a phosphate to become glucose 6-phosphate, and technically you have a different molecule. That means that you're going to have a continuous gradient. So by binding T to ABP, we are able to maintain the gradient for testosterone entry. Testosterone entry into the Sertoli cells is going to trigger the production of sperm. And obviously the sperm isn't happening inside the Sertoli cells, but rather around it, but it still is able to modulate the behavior of the sperm development. An increase in the sperm count is going to trigger the Sertoli cells to make inhibin. And so inhibin is actually going to diffuse into the bloodstream, and it will travel back and provide negative feedback inhibition of FSH and GnRH. Now, let's consider also what happens to the rest of the testosterone. So we mentioned that some of it's going to diffuse into the Sertoli cells, but not all of it. The rest of the testosterone is going to diffuse into the bloodstream. And when we get into the bloodstream, we'll have about 60% of the testosterone that binds to SHBG, which is sex hormone binding globulin. We'll have about another 38% that binds to albumin, which is just a plasma protein. And then finally, we'll have about 2% that's just free in the plasma. When we think of testosterone travel in the blood, what we're really thinking about is how we're able to exert effects on the rest of the body. So typically the testosterone will be converted to two things: estradiol, which is the primary form of estrogen, and then DHT, which is dihydrotestosterone. And between these two, we get several effects of testosterone in the rest of the body. And that would be secondary development of secondary sex characteristics, control of sex drive, increase of metabolism, and then also bone development, and especially closure of the epiphyseal plate. So basically deciding that the bones have grown to the adult length and then not allowing them to grow anymore. As you may predict, testosterone also causes negative feedback inhibition of the anterior pituitary and the hypothalamus, particularly in the context of LH and GnRH. One final thing that may be interesting in terms of the regulation of testosterone is that in order for the Sertoli cells to make sperm cells, the testosterone concentration in the Sertoli cells has to be about 100 times more than the testosterone concentration in the plasma. And so if this percentage was to decline in the Sertoli cells, then the rate of sperm production would decrease. So for those individuals who supplement testosterone, so like anabolic steroids, basically, what it will do is it will increase the plasma concentration of testosterone, and we know that the plasma concentration is what gives us our inhibition of LH and GnRH. So if we inhibit LH, then we're going to decrease the testosterone production in the Leydig cells, which means that we're going to reduce the sperm production in the seminiferous tubules. And so basically this is one suggestion for how sterility will develop in male patients who use anabolic steroids for a prolonged period of time. All right, so that was kind of a lot, but that covers our explanation of male reproduction. Hope you enjoyed it. Thanks for watching.